My name is Dr. Greg Kuchera, one of the KPC34 inventors. I'm a professor of internal medicine, section on hematology and oncology at Wake Forest School of Medicine, and I would like to introduce you to a dual mechanism targeted therapy for the treatment of cancer. KPC34 is a novel next-generation cancer treatment rationally designed to overcome multiple mechanisms of resistance currently observed with standard care leukemia and other cancer chemotherapy regimens. Nucleoside analogs such as Ara-C and gemcitabine stop malignant cell proliferation. However, getting these prodrugs into the tumor requires specific proteins, namely ENT1, to facilitate transport across the cell membrane. KPC34 bypasses this entry step because of its unique chemical properties. Once inside the cell, phospholipase C enzymatically alters KPC34 to produce two active targeted agents, a nucleoside monophosphate and a lipid. The lipid component inhibits protein kinase C, an enzyme known to stimulate cell proliferation, thus causing tumor cells to die. Meanwhile, other cellular kinases phosphorylate the nucleoside monophosphate to the active triphosphate form. This process is not dependent upon the rate-limiting enzyme deoxycytidine kinase that oftentimes becomes downregulated in resistant cancer cells. Once formed, the nucleoside triphosphate travels to the cell's nucleus, incorporates into the cellular DNA, and halts DNA synthesis, thus killing the tumor cell by a second mechanism of action. KPC34 is orally bioavailable, and it is able to cross the blood-brain barrier to kill tumor cells that invade that sanctuary site. Experiments to date have shown that KPC34 is highly effective against mice harboring leukemia cells, and it reduces tumor burden in mice with leukemia that has become resistant to commonly used nucleoside chemotherapy drugs. We are very excited about this novel agent, and we are actively pursuing a path toward a clinical trial in humans.